Welcome to today's webinar. This is the second of 3 this month, hosted by the Centre for Intelligent Design in the UK. My name is David Galloway, and I'm currently the president of the Centre for Intelligent Design in the UK. And we're a network of scientists and academics who are keen to explore the question of whether the apparent design in the universe and in life may actually be genuine rather than an illusion, as some would want to claim. We will be posting some details on our website about this ongoing webinar series, and we'll add the web details to the show notes following each webinar. Now, last Friday, we were delighted to have Dr. Mike Behe as our guest speaker, and he gave a talk on the biochemical basis for COVID-19, and that's also available on our YouTube channel. Later, I'll say a little bit more about next week's programme, but we're looking forward to welcoming Dr. Hugo van Woerden, who is a public health specialist. And by that, I mean a real trained, card-carrying public health specialist. I say that because social media these days is replete with amateur, self-styled public health experts. And Hugo will speak to the intriguing title of Beauty, Coherence, Semiotics, and Coronavirus. But today I'm delighted that we have Dr. Jonathan McLatchie as our speaker. He is an assistant professor of biology at Sackler College in Boston, Massachusetts. His undergraduate education comprised a BSc in forensic science in Glasgow, followed by a master's degree and then a subsequent master's degree before he started his PhD studies, and he completed that in molecular biology. Jonathan is an active participant in debates about design, and increasingly he's branching into various aspects of Christian apologetics. You can find him online at jonathanmclatchie.com for more of his articles and resources. Now, as before, you will be able to post your questions for discussion by using the chat facility. And after Jonathan speaks, I'm going to hand over the mic to David Williams, and David will moderate the question and discussion session, and he'll be joined by our colleague Dr. Alistair Noble, who will be there as a panelist as well. Now, we aim to conclude today's webinar somewhere between an hour and 15 minutes from now. So Jonathan, I'm delighted now to ask you to please go ahead and deliver your lecture on the molecular biology of coronaviruses. Thank you.
All right, well, thank you. It's really an honor to be here. As you mentioned, I just finished my PhD back in January at Newcastle University, studying the evolutionary origins of the eukaryotic cell cycle. My previous university studies were in molecular biology and also in evolutionary biology. And so I'm really fascinated by this question, the discipline of molecular biology and the study of microorganisms and viruses and the molecular machinery that really runs and governs the show in biology. Now today, I'm going to be talking about the molecular biology of coronaviruses, and I'm going to start by giving an introduction to what viruses actually are, and then we'll move into specifics of coronaviruses later on in particular, and look at some of the epidemics and outbreaks that have arisen as a result of coronaviruses, including, you might remember, the SARS outbreak back in 2002, 2003, and the MERS outbreak in 2012 in the Middle East, and the more recent, of course, SARS-CoV-2 pandemic, which causes COVID-19. First, though, what is a virus? Well, a virus is a genetic element that cannot replicate independently of a living host cell. And so there's a lot of debate in science about whether a virus should actually be considered alive or dead. A lot of people consider it not living because part of the definition of being alive is the ability to reproduce independently. Whereas viruses cannot reproduce on their own. Part of what makes them distinctive is that, unlike cells, they're not able to reproduce themselves unless they hijack the replication machinery of a living host cell. And so that's why they need to hijack our cells and to cause infection, because they are not able to propagate themselves otherwise. A virus particle, known as a virion, is the extracellular form of a virus, and it exists outside of the host, and it facilitates transmission from one host cell to another. And replication or reproduction occurs only upon infection or entry into the host cell. So here we see a diagram showing the comparison of naked and enveloped virus particles. And the naked virus is shown at the top, where you see the nucleic acid, which is enclosed within the viral capsid. And viruses, as we'll go on to talk about shortly, could be RNA viruses or they could be DNA viruses. And DNA stands for deoxyribonucleic acid, and RNA for ribonucleic acid. And so within this capsid is the genetic instructions of the virus. And the viral envelopes, down in the bottom here, we have this is the naked virus enclosed in this capsid down here. And around it, we have this envelope structure. And COVID-19, or the SARS-CoV-2 virus that causes COVID-19, is one of these envelope viruses. And you can see the viral glycoproteins protruding from this envelope structure, and or also known as spikes. And this is what actually gives the coronaviruses their name. Corona in Latin means crown, and because it resembles somewhat a crown, we call it coronavirus, right? And so the nucleocapsid comprises the nucleic acid and the protein in the enveloped viruses. So that's a little introduction to viral structure. So what about some of the viral components and activities? Well, there's two main types of viral infection that you might come across. One is what's called a viral interlytic infection, and that's basically where a host cell, where the host cell gets infected by a virus, and the virus replicates and destroys the host cell. It causes the cell to burst, and then it expels the virus, and it goes on to infect other cells. The other type of infection is what's called a lysogenic infection, where the host cell is genetically altered because the viral genome becomes integrated into the host genome, and it gets passed on to the future descendants of that cell, and then later it might become activated, and then and then the cell can undergo lysis and expel the virus. So what about different types of viral genomes? So as I said earlier, viral genomes might be DNA based, or they might be RNA based. And so you've got, they could comprise single-stranded DNA, or double-stranded DNA, or single-stranded RNA, or double-stranded RNA, or they could use a combination of RNA and DNA. For example, retroviruses, as their name implies, reverse transcribe their RNA back into DNA. So normally what happens is in one of our cells, for example, you have DNA, and that gets what's called transcribed into messenger RNA, and then in a eukaryotic cell like ourselves, it will be taken outside of the nucleus to the ribosome and then subsequently translated into a protein. Whereas retroviruses have RNA genomes, but those RNA genomes get reverse transcribed. So instead of going from DNA to RNA, they have an enzyme called reverse transcriptase, which reverse transcribes their RNA back into DNA so that it can be integrated into the genome of the host. And then there are also the hepadnaviruses that have small genomes of partially double-stranded and partially single-stranded circular DNA, and the genome consists of 2 strands. So you've got what's called a negative sense strand and a shorter and positive sense strand of variable length. So basically, what happens? A positive sense strand is where the virus can be the RNA sequence may be directly translated into viral proteins, whereas with a negative sense strand, it has to be converted into a positive sense strand before it can be used. So the hepadnavirus replicates through an RNA intermediate, which they transcribe back into a complementary DNA using reverse transcriptase. Okay, so viruses can also be classified on the basis of the host that they infect. So you've got bacterial viruses, which are also known as bacteriophages, which literally means bacterium eater, right? Phaga in Greek means to eat. And you've also got archaeal viruses, which infect archaea, which are a type of single cell prokaryotic cell lacking a nucleus, which tend to archaea tend to occupy extreme environments. And then you've got animal viruses, which are very extensively studied. Most work on viruses have been a lot of work has been done on animal viruses as well as bacteriophages. Plant viruses are less well studied. Not a huge amount of work has been done on plant viruses, and then there's other viruses as well. So viruses come in many shapes and sizes. Most viruses are smaller than prokaryotic cells and range from 0.02 to 0.3 micrometers. And here we see what's called the human virome, which is basically the entire population of viruses that are present in and on the human body. So on the left, we see some of the common viruses of eukaryotic cells that are found in the virome of a healthy human. And the animal viruses of the human virome include those that cause disease, such as hepatitis and SARS, as well as other more mild conditions like influenza and the common cold, which are caused by rhinoviruses and also by coronaviruses, and also latent infections like herpes virus. And the human virome is unique to an individual and is relatively stable over long periods. And the viruses of healthy humans are dominated by DNA-based viruses. Animal viruses that are typically found include anelloviruses, which are non-envelope viruses that establish persistent infections in the body early on in life, but they are not known to cause disease. Also circoviruses, which have some of the smallest known viral genomes and are typically found in poultry and pigs, which suggests that their existence in the human gastrointestinal tract might have originated from these sources. Then there are the adenoviruses, which are common respiratory viruses that are found in children with fevers and are also found in the nose and the upper respiratory tract of humans. Then there's the polyomaviruses, which are small non-envelope DNA viruses, which are quite widespread. And they can remain latent after infection, and the illnesses from those are typically occur in times of immunocompromise, such as in conditions that result in T-cell deficiency. And then there's papillomaviruses, which produce skin warts. And there's also plant viruses derived from food, which are also present in the human virome. And these are thought to cause inflammation. So in the middle figure, we see the bacteriophages, which are these viruses which infect bacteria that are found within the mucosa of the human respiratory and gastrointestinal tracts. And this is thought to protect the epithelial cells from the invasion of bacterial pathogens. And so it's thought to be the case that bacteriophages are actually our first line of defense against infection from pathogenic bacteria. And indeed, the most abundant viruses are the bacteriophages, and the large intestine contains roughly equal prokaryotic cells versus viruses. So per gram of feces, it's about 10 to the 9. And the bacteriophages are also thought to benefit the bacterial cells by transferring antibiotic resistance or metabolic genes between bacterial cells. So we also have our genome is littered with hundreds of thousands of copies of these what are called human endogenous retroviruses, which and basically our genome is like a graveyard of these past infections which have affected the primate lineage. So these are remnants of retroviral genes that have been incorporated into our genome, and they comprise about 5 to 8% of the human genome. And although most of these, so primates have been on the scene for about 50 to 55 million years, and through that time, they have incorporated infection. They've undergone infections by these retroviruses, and often they infect the somatic cells, but sometimes they infect the germline, which are the sex cells of the organism. And when they do, then they can undergo inheritance. They can be passed on to future descendants, and so they become part of our lineage for generations down the road. So most of these endogenous retroviruses that are found in our genomes are thought to be harmless, but connections have been proposed between certain endogenous retroviruses and autoimmune disorders such as rheumatoid arthritis, and with other diseases such as inflammatory bowel disease or Crohn's, as well as multiple sclerosis, amyotrophic lateral sclerosis, which results in neuron breakdown that results in the loss of function in muscles, are also associated with the expression of these endogenous retroviral elements that are found in our genome. And this is actually, I think, quite a compelling evidence for common descent among primates. So basically, the argument is that you have these insertions, and the distribution of those insertions in primate genomes forms this family tree-like organization, or a nested hierarchy distribution, if you will. And it resembles a family tree because it is a family tree, right? It represents the relationship of humans to the great apes. So humans are more closely related to chimpanzees than they are to gorillas, for example, and you can make certain predictions about the distribution of these sequences based on that hypothesis. And this is the structure of the endogenous retroviral sequence. So you've got the this up here is the retroviral sequence, and you can see at the 5' and 3' termini, or the ends of the virus, you have what are called long terminal repeat sequences. And these basically function in gene expression in the virus. And there's an enzyme called integrase, which basically inserts the virus into the host target. And the integrase is encoded by the virus itself. And because of the way that the integrase attacks the bonds of DNA, it results in this characteristic target site duplication, which is the hallmark of insertion by integrase. And you can see here the different genes of the virus have now become incorporated into the host DNA. So you've got what's called the GAG gene, the pull gene, and the envelope gene. The ENV codes for the envelope proteins of the virus. And then this can be later expressed or transcribed into messenger RNA, and now you've got the viral RNA genome. In addition to the nested hierarchical distribution of the retroviral sequences, you've also got point mutations that occur within the viral genes, right? The GAG, the POL, and the ENV genes. And those point mutations that occur within those genes also form a similar nested hierarchical distribution, which also, I think, points to common ancestry among primates. And then the 5' and 3' long terminal repeat sequences at the end, for technical reasons, have to be identical at the point of integration into the host chromosome. And so the degree of divergence between these repeat sequences can be used as a predictor of the time since integration. So you've got this kind of 3-tiered argument for common descent, which I think is one of the most compelling arguments for common descent from this distribution of retroviral sequences. However, it may be noted also that many of these endogenous retroviruses have undergone domestication. In other words, they've acquired functionality within the context of the human genome. For example, here's a paper published in Frontiers in Psychiatry titled Human Endogenous Retroviral Envelope Protein Syncytin 1 and Inflammatory Abnormalities and Neuropsychological Diseases. And basically, syncytin is a protein that tethers together the syncytiotrophoblasts, which is essential in placental formation. And the syncytin proteins seem to be encoded for by the envelope gene of retroviral sequences. Another study, and there's many more that could be cited, shows that retroviral promoters function in the human genome. So the LTR sequences, the long terminal repeat sequences on each end of the virus, can serve as promoters and enhancers within the context of the viral genome. So, so many of these retroviral sequences have acquired functions within the context of the human genome, or become domesticated, if you will. So I mentioned earlier that one of the most abundant type of virus are the bacteriophages, which are the viruses that infect bacteria. And this shows a transmission electron micrograph, kind of like a photograph, if you will, of bacteriophage T4, which infects E. coli. And it's like an alien spaceship. It's got a head and a collar and a tail and tail pins and tail fibers and a base plate. And basically the tail components function in attachment of the virion to the host and injection of nucleic acid into the host cell. And these things are really, really tiny. The head of this virus, its diameter is only about 85 nanometers, so extremely small viruses. What about some of the enzymes inside the virions? Well, we have lysozyme, which makes a hole in the cell wall to allow the nucleic acid to enter into the host cell. And it also lyses or breaks or punctures a hole in the bacterial cell to release new virions. Also neuramidases, which destroy the glycoproteins and the glycolipids, and they allow the liberation of the viruses from the cell. And then we have nucleic acid polymerases, including RNA replicases and RNA-dependent RNA polymerases. And the RNA replicases are basically RNA-dependent RNA polymerases, and the reverse transcriptase is an RNA-dependent DNA polymerase in retroviruses, which reverse transcribes the RNA into DNA to allow integration into the host chromosome. So the major difference between prokaryotic and eukaryotic viruses in nucleic acid entry in prokaryotes and variant entry in eukaryotes. So in the prokaryotes are these single-celled organisms with no nucleus, right? Eukaryotes are organisms which have a nucleus, such as our cells. And in viruses, basically the DNA or the genetic material gets injected into the host cell, but the viral head remains outside. Whereas in animal cells, such as ourselves, the virus actually enters into the host cell. So this diagram shows the replication cycle of the bacterial virus. So we have the virus, the virion here, which contains the DNA, and this is the host cell, bacterial cell. And this shows attachment, where the virion attaches to the host cell, and the viral DNA gets injected into the host cell. This is the penetration of the viral nucleic acid, and then this leads to the synthesis of the viral nucleic acid and protein. So the virus then basically hijacks the cell to translate its own genetic instruction set into proteins. And then the virus assembles itself and gets packaged into new virions, and then it will break open the cell or burst the cell, and the virions will get released and will later go on to infect other cells. So this shows the packaging of DNA into a T4 phage head. So there's these powerful motors, and there are powerful motors found not just in bacterial viruses, but also in animal viruses as well. These powerful packaging motors that basically result in this packaging, very tight packaging of the viral genetic material within the virus head. So we have a packaging motor complex for accomplishing that. And prokaryotes possess, the bacterial cells possess mechanisms to diminish viral infections. So, and this leads to a microbial arms race between viruses and bacteria. One side develops better weapons and the other side has to match those. So bacteria produce restriction endonucleases, which are enzymes which basically digest and destroy the virus particles, whereas the viruses substitute hydromethylcytosine for cytosine in the genome to basically evade the bacterium defenses. Some E. coli have evolved the altered restriction enzymes that degrade the T4 DNA. But the T4 phages modify the DNA by glycosylation instead, so they're able to evade the bacterial defenses. Okay, so and some T4 inhibit these so E. coli in response to that have developed altered restriction enzymes that recognize and degrade glycosylated T4 DNA, but some T4 bacteriophages then inhibit these modified restriction enzymes. And some E. coli evolve, in response to that, evolved uninhibited endonucleases. So what are some of these mechanisms that bacteria are using to evade viral infection? Well, I mentioned that they have several mechanisms they use. One of those is toxin-antitoxin systems. And these are basically, so a toxin-antitoxin system is basically a small genetic elements that are composed of a toxin gene and a cognate antitoxin. So the toxins of all known toxin-antitoxin systems are proteins, while the antitoxins are either proteins or non-coding RNAs. And some toxin-antitoxin systems act as antiviral factors. So if the infecting virus inhibits the host cell's transcription or its translation, which is often the case, the unstable antitoxin then disappears because it's unstable and it needs to be transcribed. And if that's disrupted, then the antitoxin is not going to be there anymore. And so the toxin is then able to exert its toxic effect to limit viral replication. The majority of toxins in toxin-antitoxin systems are used as antiviral, that are used as antiviral factors, interfere with the translation process, mostly via messenger RNA and cleavage of the transfer RNA. But prokaryotic viruses can bypass this defense system by expressing a product that's similar to the antitoxin and thereby inhibiting the host translation shutoff. Another very intriguing mechanism, which I think is a real wonder of the molecular world, is what's called CRISPR. And CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. And CRISPR regions contain short repeats of a constant DNA sequence, which are alternated with short variable DNA sequences called spacers, which correspond to pieces of viral DNA. So there are proteins known as Cas proteins, which stand for CRISPR-associated proteins, and they have endonuclease activity, and they mediate the defense against the foreign DNA and incorporate these new spacer regions. The spacer regions are basically it's basically a bacterial form of adaptive immunity, and the spacers are kind of memories of previous viral infections. So when a viral DNA is injected into the host cell, the Cas proteins of CRISPR region may recognize specific DNA sequences that are known as protospacer adjacent motifs, or PAMs. And the Cas protein cleaves the viral DNA at a locus near the PAM, which is called a protospacer, and it inserts the short DNA region into the CRISPR region of the chromosomes, where it becomes a spacer. And this allows the bacterial cell later, when it encounters the virus again, the Cas proteins are able to quickly destroy incoming DNA in an RNA-dependent way. And so that's one way that that bacteria have developed to counter viral infections. So, as I said, there's this microbial arms race between viruses and bacteria. So and there's a dynamic relationship that exists between bacteriophages and their hosts. So so sometimes the bacterial cell might alter the viral receptor sites, or modify. So they might modify the receptor or protect it with a shield, such as a capsule. but viruses can then mutate or degrade the capsule. And some temperate bacteriophages hijacked the host's toxin antitoxin systems. Bacteriophage T4 has also developed a way to evade the host cell's restriction enzymes, which are enzymes which chop up viral DNA. So this is a unique base of bacteriophage T4, which is called 5-hydroxymethylcytosine. And this base undergoes glucosylation, where there's a glucose molecule attached, and that makes the T4 DNA resistant to restriction enzyme attack. What about defense mechanisms of animal cells? Sorry, that's a typo. Defense mechanisms of animal cells, viral defense mechanisms of animal cells. So the ways that animal cells have developed defenses against incoming viruses. Well, we have a number of years ago, there was this protein discovered in human cells called TRIM21, which is a protein which basically, it used to be thought that once a virus had successfully entered the cell and evaded the cell's immune response outside the cell, then nothing could stop it. But then scientists discovered that actually the cells of our bodies have their own internal immune system. And there's a protein called TRIM21, which is searching for viral particles with antibodies protruding from the surface. Antibodies would have attached to the viruses outside of the cell before they entered the host cell. And in response to those antibodies, they attach ubiquitin molecules, which basically tag the virus for destruction by the cell shredder known as the proteasome. So there's internal immune systems found even in animal cells as well. So I want to now provide an overview of animal virus infection. So the virus binds to specific host cell receptors, which are typically used for cell-cell contact or for immune function. And different tissues and organs express different cell surface proteins, and often viruses only infect certain tissues. And entry usually occurs by fusion with the cytoplasmic membrane or by endocytosis. So here's an electron micrograph showing a rotavirus virion, showing the different stages of cell entry. So you can see this is the virus here, and it becomes engulfed into the cell. And this here is the virus actually inside the cell about to cause an infection. So the first step is uncoating of the virus, which occurs at the cytoplasmic membrane or the cytoplasm. And the viral DNA genomes enter the nucleus, and most viral RNA is converted to DNA within the nucleocapsid. And the reason for that is so that it's not recognized as foreign by the host cell and destroyed. And they bind to specific host cell receptors that are typically used for cell-cell contact or immune function. And so there's a number of effects that an animal virus might have when the cell infects. It might result in a virulent infection or lysis of the host cell, which is most common. Or it might result in a latent infection where the viral DNA exists in the host genome and virions are not produced, and the host cell is unharmed unless and until virulence is triggered. It may also result in a persistent infection, which is where you have release of the virions from the host cell by budding that doesn't result in cell lysis, where the infected cell remains alive and continues to produce the virus. Or a transformation, which is basically the conversion of a normal cell into a tumor cell. So this again shows the structure of a retrovirus and its function. So retroviruses, as I said earlier, are RNA viruses that replicate through a DNA intermediate. So a well-known example is the human immunodeficiency virus, or HIV. And they contain a reverse transcriptase, which copies information from RNA to DNA. And they also contain integrase, which is responsible for integrating the the genetic instructions of the virus into the host chromosome, and also protease, which is responsible for basically chopping up the is a single polypeptide that's produced, that's translated, and the protease basically cuts it up to produce the different viral proteins. And these are enveloped, so you can see the envelope that surrounds the viral capsid. So retroviruses have a unique genome. So they have two RNA molecules, and they contain specific genes, GAG, POL, and ENV that I discussed earlier. So the GAG gene encodes the structural proteins, and the POL encodes the reverse transcriptase and integrase. POL stands for polymerase, and then ENV, which stands for envelope, encodes the envelope proteins of the virus. So this shows the life cycle of a retrovirus. So you've got entry and uncoating of the retrovirus here, and then you've got the reverse transcriptase activity and the entry of the viral DNA into the nucleus and integration into the host chromosome. And the transcription by the host RNA polymerase forms the viral messenger RNA and genome copies. And then finally, the translation of the messenger RNA forms the viral proteins, and the new nucleocapsid are assembled, and they are released by budding. And so there's the release of the virus. So what theories have been proposed to explain where viruses came from? You might remember from Dr. Behe's presentation last week, no one's quite sure exactly the origins of viruses, but there are a number of hypotheses that have been put forward in the literature. It's thought that viruses may have arisen after cells. That's one hypothesis. And there's some evidence that's taken to suggest this. For example, all known viruses require a host cell for their replication, and the remnants of cells that replicate they are remnants of cells that replicate with the help of a live cell. Viruses, as part of the RNA world, may have played a role in the transition from RNA to DNA. And that's one hypothesis that's put forward, although that will depend on the traction you give to the hypothesis of the RNA world. And I think there's significant problems with the RNA world scenario, which if you want to ask about in the Q&A, I'm happy to discuss. And it's thought that viruses may have been the first forms of life. In other words, they were pre-cellular. So that's another hypothesis that's put forward. Now, the next question that might arise is, well, why did viruses appear on an evolutionary scheme of things? Well, one hypothesis is that they arose as a mechanism to quickly move genes from one cell to another. So viruses have an extracellular form that protects the nucleic acids inside them. And so perhaps the argument goes they were selected as a method of improving the genetic diversity of their hosts by mediating the transfer of genes between cells. And latent viruses don't kill their host cell, and perhaps the earliest viruses were primarily latent, and the lytic abilities of viruses developed much later on to allow them to access new hosts more quickly. So the proteomic evidence suggests that viruses originated from ancient cells containing segmented RNA genomes, and they've existed prior to the last universal common ancestor. So in the RNA world era, and that there was, so the hypothesis is that there was an evolutionary pressure for a reduction in the genomic and compartment size. And so over time, this removed the cellular nature of these viral cells, which left only a protein shell to protect the genome. And that reduction would have resulted in the viral particles being dependent on cellular hosts for their replication. And so that's why they require infection of host cells in order to replicate. Okay, so there's also a popular hypothesis that some RNA viruses evolved DNA genomes as a way of protecting their genomes from cellular ribonucleases that destroy foreign RNA in the cell. And the reverse transcriptase enzyme is thought to have been important for the transition from the RNA to the DNA world. And it's thought that DNA viruses subsequently infected the ancestors of the cellular domains, and so that's one hypothesis that's put forward. Polioviruses are one of the smallest viruses, and they have an only 30 nanometer icosahedral structure, and the replication takes place in the host cytoplasm, and they attach to a specific receptor on the host cell surface and enter into the cell, and they get uncoated, and the genomic RNA is attached to the ribosomes and translated to yield a polyprotein down here, and then this polyprotein will be cleaved by protease into these different viral proteins. So what then about coronaviruses? So coronaviruses are a whole family of viruses, and these are single-stranded RNA viruses. And like the poliovirus, they replicate in the cytoplasm, but they have a larger size than polioviruses, and they don't have the polyprotein formation and cleavage that you find in polioviruses. And coronaviruses cause respiratory infections in humans as well as in other animals, and about 15% of common colds that we experience result from coronaviruses, and most common colds result from rhinoviruses, but about 15% arise from coronaviruses. And there have been a number of prolific outbreaks of coronaviruses, including the SARS-CoV-1 pandemic in 2002, 2003, and also the MERS epidemic, which or outbreak, I should say, which arose in the Middle East in 2012. And of course, the more recent COVID-19 or SARS-CoV-2 pandemic. So here we see the coronavirus, and as you can see, it contains these spikes or these glycoproteins, and that's how it gets its name as being coronavirus, because it looks like a crown. You can see how it infects the cell and results in, it basically hijacks the cell's machinery, and then it gets expelled from the cell and goes off to infect other cells. So as I said, the common cold, about 50% of common colds are caused by coronaviruses, and about 40 to 50% are caused by rhinoviruses. And it's got a seasonal incidence because they don't survive so well in the cold temperatures, and so that's why the colds are more prevalent during the winter months than in the summer. So the SARS, the original SARS pandemic, or the SARS-CoV-1, occurred 2002, 2004. And fortunately, it didn't turn into a pandemic like SARS-CoV-2, but it did result in a severe outbreak, which was very scary, especially since so in June 2003, the incidence of the SARS-CoV-1 was 8,422 cases. And the case fatality rate, right, so that's the number of deaths over the number of people infected, was 11%, which is much higher than the 1 or 2% case fatality rate for the SARS-CoV-2. So that would be much more of a scary SARS virus than the one that we're currently encountering. And so in late 2017, Chinese scientists traced the virus through the intermediary of Asian palm civets to cave-dwelling horseshoe bats in Yunnan. And the case fatality rate basically ranges from 0 to 50%, depending on the age group of the patient. So patients who were under 24 were least likely to die, about less than 1% case fatality rate, whereas those who were 65 or older were the most likely to die, with a case fatality rate of over 55%. And what's interesting about these coronavirus outbreaks is that it results in significantly more deaths of males and females. And no one's really quite sure why that is. It's the case with the new SARS-CoV-2 pandemic as well, and also with MERS, which I'll discuss in a minute, the Middle Eastern Respiratory Syndrome. So Middle Eastern Respiratory Syndrome was an outbreak that occurred in 2012 in the Middle East. And the typical symptoms of MERS include fever, cough, diarrhea, and shortness of breath, similar to the COVID-19. And about 2,500 cases have been reported, and about 35% of those who are diagnosed with the disease die from it. So also very, very scary. So then this brings us to today, which is the coronavirus disease 2019, or COVID-19, which results from the SARS-CoV-2 virus. And the common symptoms include, again, fever, cough, fatigue, shortness of breath, and loss of smell and taste is actually reported in some patients as well. Okay, so what about the source of the SARS-CoV-2 pandemic? Well, scientists have been able to show that it's about 96% identical at the whole genome level to other bat coronavirus samples. Also, there's only one amino acid difference in the binding domain of the S protein between the coronavirus from pangolins and those from humans. But whole genome comparison to date found that at most 92% of genetic material is shared between pangolin coronavirus and SARS-CoV-2, which is insufficient to prove pangolins to be the intermediate host. So it looks like it's derived from bats, but whether there was an intermediate host and what the identity of that intermediate host is not certain at this point. So how does a SARS-CoV-2 infection occur? Well, this is a diagram showing the structure of the SARS-CoV-2. You can see it's an envelope virus, and these are the spike proteins which bind or dock to this docking site, which is known as ACE2, which stands for angiotensin converting enzyme, or ACE2, and it's and so that's the binding site of the SARS-CoV-2 and its way of getting into the cell. Now, what is the people have actually measured where the ACE2 docking site is expressed. And interestingly, it's highest in the nose, right? It seems to be highest in the nose. And people thought in the beginning that you needed to get these viral particles way down into the deep part of your alveoli down here. But that turns out actually to not be true. So you don't actually need to worry about the small particles that the N95 masks are really good at excluding, but the homemade masks are not good at excluding. So wearing a mask is a good defense against getting the larger viral particles in your nose. And so that's, I think, a good reason why people should be wearing masks. In fact, this study that was published quite recently on professional homemade face masks reduced exposure to respiratory infections among the general population. They showed actually just how effective masks are in preventing infection. So this shows this is the efficacy of protecting yourself, or inward protection. So in wearing a tea cloth or homemade mask, about 33 in 100 particles were leaked through the mask. So it stopped most of the particles getting through. A surgical mask, only 25 of 100 got through. And with the N95, 1 in 100 got through. Whereas protecting others, or outward protection, about 90 particles leaked into the environment. Because when you're sneezing, then the particles are moving at very high velocity, so they're more likely to get out of the mask. So about 90% of particles got out of the mask, 50% that's for the tea cloth or the homemade one. For a surgical mask, about 50% did, and for the N95, the professional one, about 30% did. And so wearing a mask is actually far more beneficial for protecting yourself rather than for protecting others, contrary to what was thought at the beginning as well. So let me conclude then by asking this question that everyone seems to be asking at the moment. Was the SARS-CoV-2 virus man-made? And in fact, it turns out that nearly 3 in 10 Americans believe COVID-19 was made in a lab, according to Pew Research. Well, certainly it couldn't have been created in the lab from scratch because there's just simply not enough knowledge about how to create a new virus that can cause the devastation of SARS-CoV-2. So I think that is just ruled out. But could SARS-CoV-2 have arisen by modification of an already existing virus? Well, when scientists have analyzed the genome and the insertions and mutations that have occurred within the SARS-CoV-2 genome, there are no genetic insertions that have occurred that aren't also identified in nature. So there are insertions and deletions going on that aren't already occurring in nature. Furthermore, so the next question is what if what if they mutated an already existing virus and tried to make it become more pathogenic or make it become more transmissible or something like that? Well, there's a technique known as serial passage, which basically works in a similar way to selective breeding, and scientists are able to some extent mimic the process of evolution. And so by forcing the virus to mutate over and over again, they can potentially create a new, different form of the virus. And that can be used to weaken the virus, which is how we create some vaccines, or it could be used to strengthen the virus by making it, by rendering it more transmissible. But for that to really work, the existing virus would have to be very, very similar to the new virus. And in fact, the backbone of SARS-CoV-2 is unique, differing quite markedly from other coronaviruses. So that seems to counter some evidence against that idea. The first SARS actually only has the SARS-CoV-1 virus only has a 79% sequence match to SARS-CoV-2, which is not a particularly good sequence match. And the best candidate is the RATG13, which is a bat coronavirus with 96% similarity. But that's actually quite massive for viruses. So the genomes actually vary by about 800 nucleotides, which is about 50 years of natural evolution. And a virus such as SARS-CoV-2 has about 30,000 nucleotides in its entire genome. So that's quite a lot. So it's and the reason why the viruses would have to be very, very similar is because the process only speeds up viral evolution, and it has a limit. So it's not possible to direct mutations into a completely different form. So what about the hypothesis that SARS-CoV-2 is actually a known virus that people in China were working on in the laboratory and that leaked from the lab? Well, I think this is a possible hypothesis, but I think it's not a very likely one. So and the reason for that is, one of the reasons for that is that the virus was not sequenced prior to January 2020, and it would seem that if it was a known virus, it would have been sequenced prior to that. And it's also statistically improbable, given that there are many more people that have encounters with animals, and of course, SARS-CoV-2 is a zoonotic infection, or is widely thought to be a zoonotic infection. That is an infection acquired from animals. And there's only a small handful of scientists working with coronaviruses in the labs. So it's also statistically unlikely. So while that conjecture is at least possible, I don't think it's very probable. So I hope I've given you a short introduction to virology and coronaviruses in particular, and hopefully also what I've discussed encourages you to wear a mask and shows the importance of wearing a mask for your own health, because it has been shown to reduce the number of viral particles entering your body. So I'll close with that, and I'm happy to take any questions. Thank you.
Thanks ever so much, Jonathan. Really interesting talk. I'm sitting outside, so I don't need to wear a mask. Is that right?
In proximity to others.
I'm going to jump straight in with the design question. Just before I do, anyone that wants to ask a question, some have emailed direct, others have done it on the YouTube live feed. So if you want to type a question there, and I will hopefully get around to asking it. But straight in with the design question, Jonathan, you mentioned, or you addressed the questions of when viruses had appeared, why they appeared, and how they appeared. And you talked about various hypotheses and what has been suggested. You didn't actually say whether you thought any of those were correct from an evolutionary standpoint, and if you do, which one, and if not, are you saying they were designed?
Yeah, so I should have said these hypotheses are very speculative, right? So there's not a lot of empirical support, which is why Behe mentioned last time that no one's really quite sure where viruses arose from. These are a few of the hypotheses that have been floated in the literature. And it's of course going to depend on the amount of credence you give to the RNA world scenario and things like that. And I think there are significant problems with the RNA world scenario. For example, RNA, the way this differs from DNA is in its possession of an extra 2' hydroxyl group. So hydroxyl group is an OH group, which it has an extra one of those where DNA doesn't, which makes it more reactive and less stable than DNA. And also RNA is single-stranded generally rather than double-stranded, which makes it less stable as well. So the RNA world scenario seems to me to be implausible for those and other reasons. And I've written about this at the Evolution News and Science Today blog, so people can check out my author page if they want more details. Stephen Meyer also covers it in his book Signature in the Cell. So yeah, these are very speculative scenarios. I don't buy the RNA world scenario. I also don't really buy the naturalistic evolution theories.
So would you say therefore that it's by default that you're saying they're designed, or are you saying there's evidence for design in viruses?
I think some of the viral machinery is very impressive. Such as I talked, for example, about the motor that packages the viral genetic instruction set into the viral capsid, which I think seems to point to design, so I think there's positive evidence of design in terms of the viral machinery, the way that they are able to gain entry into the cell by carrying counterfeit paths and that type of thing.
Okay, you talked about the evidence, strong evidence for common ancestry. One of the questions, or a question is, you talked about them being present, HERVs, I'm talking about human endogenous retrovirus. Yeah. You talked about them being present in primates. Are they, one question, are they present in other animals as well, yeah, or just in primates?
Yeah, they're found in all animals, yeah.
And so is there a continuous lineage back beyond primates that can give further information about descent?
Yeah, there are. There are phylogenetic trees that have been constructed using retroviral sequences. I think the evidence is especially strong for primate common ancestry from those sequences, which is why I talked about primate ancestry.
And so, is there evidence for when they first appear? And if so, again, are you saying they appear by design or by some evolutionary mechanism?
When retroviruses appear?
Yes.
Yeah, so retroviruses appear to be very, very ancient, going back billions of years.
Okay. You talked about the replication cycle of a virus, and you showed the system, if I can call it that. One of the question that's coming on that is, how would a system like that, the replication system, be explained in evolutionary stages or steps?
Sorry, the replication system for, sorry, say again?
The replication system, replication cycle of a virus. There was a slide or two on that. The question is, how would a system, as to how a virus replicates, how would that system of replication evolve in gradual steps?
Yeah, I reject gradualism. So I don't think that there is a plausible mechanism for the evolution of these systems.
So again, does that mean the default is design?
I think that when we see molecular machinery and well-matched interacting components, molecular motors, and that type of thing, information-rich systems, we habitually associate that with design. So it's not that the default is design, but design is the best explanation or the most causally adequate scenario to account for them.
Another question on the YouTube feed is in the context of what you're just talking about being present in not just all primates, but all animals, evidence for common descent, could it not also represent evidence for common design?
Yeah, so the argument is similarity is not necessarily evidence of descent because you can take it as evidence for common design. In principle, yes, but the argument for common descent more relates to the distribution of similarities and differences, so them forming this nested hierarchical distribution. And the fact that these, so I gave the example in the presentation of the endogenous retroviruses, where you have this distribution which forms a nested hierarchy or a family tree, and these are genuine inserts, and we know this because of the characteristic target site duplication that they leave behind. But so they are genuine inserts, but they form the exact pattern you would expect on the hypothesis of common ancestry.
Okay, one more question on common descent, and then I'm going to move to an area of philosophy or some philosophical implications. On common descent, a question that's come in is you obviously believe that primates and humans have a common ancestor. Or maybe you believe in.
I think it's the best explanation of the evidence currently. There is certain. I don't think it's as conclusive as many people think, but I think I would tend to interpret the evidence that way.
And the question went on: is that normal belief for ID or proponents of ID? I suppose that question means.
There are people in both camps on that. Mike Behe, who presented last week, subscribes to common ancestry. I'm not so sold on universal common ancestry. I think that there's certainly evidence for limited levels of common ancestry.
Okay, if I can move on to the couple of questions or a few questions on the philosophical implications, it may be that Alistair Noble can join in with some comments here as well. But first question is this: if there is need for a designer, does that not imply malevolence on the part of that agent or that designer?
Yeah, so that relates to theodicy, which is a theological question. So there's a strictly scientific question, then there's a more theological question. So the question is, I mean, if you're a Christian theist like myself, then you want to ask the question: does the Christian worldview have the explanatory resources to account for that? But that's, I think, a question that goes beyond the scientific evidence, and so I can give theological responses to that, but that's, I think, independent of the scientific evidence.
We obviously, at the moment, at the forefront of our minds is coronavirus, which we would view as a bad virus, if I can put it in simple terms. Is there such a thing as a good virus?
Yeah, so I mentioned earlier the fact that bacteriophages, which are viruses which infect bacteria, are possibly our first line of defense against bacteria, because they actually infect bacterial cells and leave our cells alone. So I guess in that sense you could say so.
So what would be the theistic view as to good and bad viruses?
I think it's a good question. From a theological point of view, one could argue, and I've heard some people argue this way, that perhaps there are two intelligent designers, and you've also got you've got God as an intelligent designer. You've also got Satan as an intelligent designer. I think that's a view that one can accommodate within a theological framework.
Okay, there's another question on the YouTube feed. I'll read it. Now it says, philosophically speaking, is there not an argument from what is known about viruses and presented this evening that they are linked to the problem of evil in the world?
Yeah, so from a theological point of view, one can say that, this gets into kind of controversies about the nature of sin and the nature of the fall and the extent to which the world has been marred by sin. And from a biblical point of view, I don't actually think that Scripture says that natural evil in the world is the result of human sin. I think you can argue that there's another fall prior to that, of Satan, and you have at the time of Adam and Eve being in the Garden of Eden, you already have satanic influence on the animals. For example, it mentions that the serpent was the craftiest of the beasts of the field, for example. So I think there are ways of understanding that.
Okay, another question from YouTube, moving away from the philosophy for a second. And it's this: what are the theories about how coronaviruses transmit from one species to another, e.g. bats to humans?
Right. So typically, so these are called zoonotic infections, which is a trans-species infection. And these can happen when someone gets too close to an animal, and the particular virus that's associated with that animal has undergone a mutation. So it's resulted in a new strain of that virus. And that makes it it's changed its receptors in such a way that it's changed its glycoproteins in such a way that it's now more compatible with the receptors in our cells.
Okay, and another question: do coronaviruses have non-genetic information in them, such as distribution, expression of membrane proteins?
Non-genetic information. Sorry,
I'm reading word for word. Do coronaviruses have non-genetic information in them, such as distribution, expression of membrane proteins?
No, viruses aren't cells, so they don't have a plasma membrane.
Okay. Widening it out to ID, more general ID, the question that's coming is: was Dembski wrong about specified complexity as an argument for design? I think it might be useful if you first of all briefly explain the specified complexity argument before saying if you think he was wrong.
So specified complexity is the idea that you have that the two criteria have to be met in order to warrant an inference to design. One of them is complexity, and the other is specification. So complexity relates to the improbability of something happening by chance, and specification relates to conformity to some independently given pattern. So complexity always relates to the probabilistic resources at one's disposal, of course. And the short answer is no. I don't think he was wrong. I think that that is a good set of criteria for inferring design.
Okay, now I promised last week there were two questions we didn't get around to, so I'm going to chuck at least one of those in now, if I may. And it's this: can you explain the mechanism of citrate utilization in Lenski's experiment? Do you think this leads directly to loss of fitness in these populations, or is this due to more downstream effects? Now, that's the question. Let me break it down first of all. First of all, can you briefly tell us what Lenski's experiment was, and then move on to the mechanism of citrate utilization?
Yeah, so Richard Lenski is a biologist at Michigan State University, and he's most famous for his long-term laboratory evolution experiments done with Escherichia coli. And basically, he's been working with his lines of E. coli for many decades now, and he's been trying to observe what changes occur in a population of E. coli over the years that he's been working with them in the lab, because E. coli and other bacterial cells have a much more rapid generation turnover time and much more massive population sizes and higher mutation rates. You can basically get equivalent to millions of years of human evolution or animal evolution because of the rate at which bacterial cells divide and so forth. And one of the changes that he's kind of championed is or made known is the ability of one line of E. coli to be able to metabolize citrate, which it wasn't able to do before. And there's a couple of possibilities of how that might have occurred. One is that it resulted from a loss of regulation of an already existing citrate transporter, or loss of specificity. And indeed, it seems that seems to be the case. In fact, Dr. Scott Minnick published a paper a few years ago. He's a biologist at University of Idaho, and he and his lab basically showed that under certain conditions, they could get the citrate metabolism to evolve very, very quickly within a period of days rather than years. And so they argued that actually it's quite an easy innovation to occur, and most likely results from a loss of function mutation, such as a loss in specificity or a loss in regulation.
Okay, well, Jonathan, I've got one more question for you. Before I come to that, thank you ever so much for taking the time to give this talk this evening. It's been hugely informative, and thank you for taking the time to answer the questions as well. So I'll hand back to David Galloway after this question, but a thank you for me. And the final question is this: what's the connection between mutations and an allele? Are the alleles spoken in biology formed by mutation? And if so, why so few alleles?
Yeah, so an allele is a gene variant. So it's just when you have two versions of a gene which have slightly different sequences, then that's called an allele. And a mutant or mutation gives rise to a new allele. So I think was that the whole question?
I think so. Yes.
Yeah.
David, is it too late for me to ask a point of Jonathan?
No, go for it, Alistair.
Jonathan, I've got a specific and a general question. My specific question is, when you were talking about our DNA being a graveyard for some of the RNA or DNA of viruses, an iconic argument about evolution is that a large part of our DNA is junk. And then the ENCODE project suggested that most of our DNA was active. Are you suggesting that the viral presence corrupts our DNA in any way, or does it collide with what the ENCODE project has found about DNA?
Yeah, so as I mentioned, there's been an association between some of these sequences and some diseases, and there's also been autoimmune diseases, as the word I was looking for, and there've also been examples where, and I mentioned this in my presentation, where these retroviral sequences have become domesticated within the context of our genome, such as in the case of syncytin, for example, and giving rise to promoters and enhancers. And there's actually a wealth of literature on the functionality of endogenous retroviral sequences. But although these sequences do seem to be functional, or many of them seem to be functional within the context of the human genome, these do, to me, seem to be inserts rather than something that was originally present. And so I think that even though they are functional, I think that they are still they still provide evidence for common descent.
And my general question is to do with good viruses and the benefits of viruses. Now, I get a sense you point to bacteriophages as being the first line of defense, but last week Michael Behe was suggesting that viruses have a widespread function in the biosphere. Are we struggling to find examples, positive examples, of what viruses do constructively rather than destructively?
I mentioned bacteriophages, which infect bacterial cells and are kind of one branch of our immune system. There's not. I mean, most viruses are pathogenic. I mean, they need to be pathogenic because they have to hijack our replication machinery in order to replicate themselves. So certainly, more viruses are unfriendly than ones that are friendly.
Thanks, Jonathan.
All right. Thank you.
David Galloway.
Yes, well, thank you very much. Thanks to everyone who's been participating this evening. I'm now reeling with various thoughts in my head about good and bad viruses and helpful and unhelpful viruses. I have to go and have a think about that. But let me just take this opportunity to thank Jonathan for his talk and David and Alistair for participating in the discussion, moderating the questions that came in from the YouTube channel and elsewhere. Let me also, just as we wind up today, remind you about the website, which is c4id.org.uk. And again, we'll put a link to that during the next day or two onto the show notes for today's presentation. So, very interesting that you've been given the opportunity to explore something tonight which probably for many people not deeply conversant with the language of molecular biology perhaps found the detail a little bit high speed and overwhelming. Well, you can always go back and check it out. And indeed, when you connect to our website, you'll find some resources there calling into question the purely naturalistic understanding of the origin and design of life, just as tonight we've heard a bit of a challenge to the purely naturalistic understanding of the origin of viral reality. There are also other resources there which explore different aspects of design and biology and in cosmology, and still others plough into the philosophical implications that we touched on already. So as we wind up, let me just finally take this opportunity to thank everyone for connecting and for joining. I hope you found it useful and interesting. And we look forward very much to welcoming Dr. Hugo van Woerden next week, same time. So do plan to join us then. So I'm on the island of Arran off the west coast of Scotland. David Williams is in rural Wales by the looks of things. Jonathan's in Boston, Massachusetts, and Alistair's in Glasgow. And so from all of us, good day, and we look forward to connecting again next week. Goodbye for now.